So for several years now, we've run something called the BioNewsRound Award at Genesis, recognizing those that have done good things and communicated good things, because that just builds confidence with everybody else. A little bit different this year is the fact it's the first time we've had the BioNewsRound people presenting at Genesis, because we get feedback saying, oh, I'd like to know more about them. So here's the chance to have a snapshot through the window of why they're the finalists, and that's going to be moderated through by one of our newest colleagues. It's been 3 years, but it doesn't felt like it. So welcome to the Genesis stage for the first time, Jasmine.
Thank you, Tony, and hello everyone. I hope you're enjoying your Genesis conference so far. I'm Jasmine Bannister, and I'm the Member Engagement Manager at One Nucleus. It is my pleasure to guide you through the next part of our program, where we'll be hearing from the 6 finalists of the BioNewsRound Award. They'll each be giving just a very brief pitch on why they believe they should win. So with that, I'm delighted to welcome Neil Miller from NRG Therapeutics for being awarded their second $500,000 grant from the Michael J. Fox Foundation to further progress their Parkinson's program. So with that, over to you, Neil. Thank you.
Thank you. Good morning, everyone. I'm Neil Miller. I'm CEO of NRG Therapeutics. We're a neuroscience company. We're interested in mitochondrial biology, and in particular, we're interested in the permeability transition pore, for which we feel the science has now emerged to make that a compelling mechanism to go after in ALS, or motor neuron disease, and in Parkinson's disease. What differentiates us are 2 things. First of all, we've discovered a protein that is essential for the opening of the pore, and that's a real breakthrough in the field. And secondly, we have the only orally bioavailable and CNS penetrant small molecule inhibitors of the pore. So we're uniquely positioned now to test this preclinically and take this into the clinic. In the interest of time, I won't go through the team on the left, but you may recognize some of the mugshots. In terms of our history, the company was founded in 2019. We were seed funded by the charity Parkinson's UK, and then we secured a 16 million Series A round we closed last summer that was led by Omega funds. And that set us up to advance our molecules to the point of clinic entry. We've also been very successful in raising non-dilutive money on the way, and we secured a large BMC grant from Innovate UK that was extremely helpful. And we've secured 2 grants from Michael J. Fox. And it's the second of those that I guess is the reason I'm standing here today and I will talk about. Our hypothesis is that mitochondrial dysfunction, which is common in all neurodegenerative diseases, is driven partly or wholly by a consequence of the pathological proteins in those diseases. So in ALS, that protein is TDP43, which mislocalizes into mitochondria. It induces the release of mitochondrial DNA through the MPTP. That initiates the innate immune system, and you observe the downstream motor neuron death. And in Parkinson's disease, the protein's oligomeric synuclein, which co-localizes to mitochondria. It sensitizes the pore, and we believe in substantia nigra neurons. That helps us understand why they die under the high calcium load that they're exposed to. So that's our hypothesis. Let me tell you a little bit about mitochondria. I think important to remember they're not just batteries of cells. We're particularly interested in their ability to regulate and store intracellular calcium, and that story is particularly relevant in Parkinson's. And we're increasingly interested in their ability to talk to the innate immune system, and that science is, we feel, very important in the story around ALS. The pore itself is a high conductance channel. It sits on the inner mitochondrial membrane. It's activated by a number of cellular stresses, best characterized with calcium reactive oxygen species. And once activated, there's a loss in membrane potential. The pore opens and a release of solutes. And large macromolecules come out like mitochondrial DNA and cell death factors that initiate downstream cell death. So there's a compelling rationale for going after this mechanism. The challenge has been, A, a lack of understanding of what forms the pore. The only known validated regulator is a protein called cyclophilin D, and many groups have shown if you take mitochondria that are deficient in cyclophilin D, they're resistant to pore opening, and through genetic or pharmacological inhibition of CYP-D, you can rescue mitochondria, you can protect cells, and you can extend survival in animal models. So it's a compelling pathway to go after in neurodegeneration. The challenge has been chemical matter. So cyclophilin D is a peptidyl prolyl isomerase, and the only known inhibitors of that protein are large macrocyclic peptides, cyclosporinase, the prototypical inhibitor, that just do not get into the CNS. And we have solved both of those problems. And the first of those problems was identifying or learning more about the composition of the pore. And this is work that was funded by the Fox Foundation. What I'm showing here, so we identified what I'll call protein A through a classic chemoproteomic experiment. And what I'm showing here on the left-hand graph is an experiment which we take isolated mitochondria, we add calcium to them, they absorb to the point they can absorb no more, and the pore opens. And you can see that's in the control experiment in the black and purple line. You see the opening after 2 or 3 additions of calcium. In blue is the effect when we knock out protein A in mitochondria in mice. And so we see a full blockade of the pore. And we've replicated that experiment with a small molecule inhibitor shown on the right-hand side. So again, you see in black the control experiment. In the presence of one of our small molecules, you see pronounced prolonged blockade that's equivalent to the knockout. What I can tell you about protein A is that knockout animals are healthy and viable with no overt phenotype, and there are also a small number of humans around with loss of function alleles in protein A that are reported to be healthy. So we don't believe we're targeting a pathway or a mechanism that's inherently unsafe. I can also tell you that protein A is a member of a family that industry is super excited about, actively working on, including in CNS indications, but no one has worked on this protein, or at least no one has understood its full or its real function, and that's what we've discovered. And so our second award from Michael J. Fox is to really understand the role of protein A in mitochondrial biology and how that relates to pathology, in particular in Parkinson's disease. Let me just finish by concluding with, this is our pipeline. We have 2 chemical series in mid-stage lead optimization. They're structurally very distinct, they're potent, selective, and they both act through protein A. They're orally bioavailable, they're CNS penetrant, and we now have data showing they're neuroprotective, they reduce neurofilament light chain in seizure models, and those lead molecules now being progressed into some efficacy studies in ALS and in Parkinson's, and we're running some non-GLP toxicology in parallel, and so we hope to be candidate selecting toward the end of this year, early next year, and moving to the clinic by the end of 24. And I'll stop there. Thank you.
Thank you, Neil. Next up, we have Roberto Zanchi, who is representing the Quadram Institute and Oxford Nanopore for the news of the World Health Organization supporting the use of targeted sequencing to detect drug resistance in tuberculosis. Over to you, Roberto.
Thank you. It's a pleasure to be here today, and I would like to thank the Genesis organizers for the opportunity to allow me to tell you how, in the last couple of years, we've been leveraging each other's capabilities and maximize the impact for life science innovation. So for those that are not familiar with the Quadram Institute, we are a cutting-edge research institute based on the Norwich Research Park. It's a unique cluster of excellence. So together with us, there are other 2 BBSRC-funded institutes on the park, and we heavily collaborate with each other and leverage each other's capabilities together with the hospital and the University of East Anglia. So we are all closely co-located, and of course we collaborate with companies on the park as well. So the Quadram Institute brings under one roof both clinical and fundamental biology expertise. It is a state-of-the-art facility that represented £75 million investment by BBSRC. And as you can see here, we really have a way to go from the bench to the bedside and vice versa. So we can really generate that innovation and translate it. And within these state-of-the-art facilities, we have a space for collaborative innovation where we can incubate spin-outs, as well as support and foster those collaboration with companies such as Oxford Nanopore that help us to bring our innovations to patients and to the real world. So I'm sure all of you know about the need and the challenge of rising antimicrobial resistance, but let me just remind you and emphasize the case for tuberculosis. So tuberculosis is really a major cause of morbidity and mortality worldwide. In 2021, over 10 million people developed active TB that leads to over 1.5 million deaths every year. Among infectious diseases, this is only dwarfed by COVID-19 in 2021. So only COVID-19 caused greater mortality. It is a notoriously difficult disease to treat. It takes 4 to 6 months of antibiotic, of a combination of antibiotic in order to overcome the disease. The resistance to first-line therapy is increasing and currently stands about to 10% of all cases. And when there is resistance, often that requires treatment with at least 6 anti-tuberculosis drugs. So as you can see, because of this, it leads to a potential for continuous transmission of the disease within the community because it takes so long to treat the disease. And also, in absence of diagnostics, often you won't see a response for quite some time, which compounds this transmission. And unfortunately, the picture you see here is not relegated to the archive of medicine. Unfortunately, in the settings where the burden of TB is highest, this radiology still underpins a lot of diagnosis. It is rapid, relatively, but it doesn't inform, and it doesn't provide any information about drug resistance. And in fact, while testing for resistance is recommended, unfortunately, there were no WHO-recommended rapid diagnostics that can inform really the treatment decision in a single test. And that's where this is the stark backdrop of our collaboration with Oxford Nanopore. So in 2019, Professor Justin O'Grady's group at the Quadram developed an in vitro test that can diagnose and can test up to 16 can diagnose the resistance to up to 16 drugs. And the partnership with Oxford Nanopore really provided us with the right platform to bring it to the patients because it enables the performance of the test in under 5 hours, and it has a great portability. And again, in the settings where TB is prevalent, this is key because the patients have to travel, and often they need to be treated when they come to the clinic, and you might not see those patients for many months after that. So also the partnership with Oxford Nanopore enabled the clinical trials in the field, also together with FIND, and this really led to the center of the announcement that was shortlisted for the award, whereby WHO endorsed the test. And this is so important because getting a diagnostic through regulatory approval is often half of the challenge. Actually changing clinical practice is the other half, and getting that adoption clinically. And this endorsement by WHO is key, and it will really drive the implementation of this testing in the field. And so I hope I have persuaded you to vote for us, because I think this is a fantastic case, an example of how industry and academia collaboration can really leverage each other expertise in life science innovation and can bring real tangible benefit to patients. And I think this is a great example. I hope you can vote for us.
Thank you, Roberto. Moving on, I'm delighted to welcome Kat Arney from First Create the Media, who will be pitching on behalf of Etcembly for the reveal of the world's first generative AI-designed bispecific T-cell engager with picomolar affinity. Thank you. Over to you, Kat.
Thank you. Super. So yeah, hello. Thank you very much to the One Nucleus team for nominating our story for the Genesis BioNewsRound Award. I am the founder and chief creative officer of First Create the Media. We're a science communication, strategy and content agency. We work with amazing companies in the life science, and we've been working with Etcembly over the past year as they're coming out of stealth mode and starting to share their story with the world. So in the spirit of the news round, I'm going to talk not only about the good that they do, but also how we have communicated this story to the world. Now, Etcembly is an Oxford-based biotech founded by these two amazing people. That's Michelle Teng and Jacob Hurst. And they really bring together cutting-edge immunology, that's Michelle's domain, with cutting-edge AI, and that's Jacob's domain. And what they're trying to do is create next-generation immunotherapies that are going to transform patients' lives. So this is how they do it. They're using generative AI and structural modeling to develop a pipeline of high-value TCR-based immune engagers. So basically, similar to antibody-based immune engagers, these use T-cell receptors to redirect the patient's own immune system to tackle diseases such as cancer and autoimmune conditions. The problem is that unlike antibodies, it's very, very difficult to screen for TCRs against a specific target. This is because they only recognize antigens that are bound in the HLA complex on the surface of their target cells. Now, furthermore, there's another problem. TCR binding is inherently weak, and there's also a lot of cross-reactivity with healthy tissues. So this means that if you want to find a TCR for a specific target, that's not enough. You then need to do a lot of engineering to make it high affinity and to eliminate cross-reactivity if it's going to be effective as a therapeutic. Now, to solve this problem, Etcembly have developed Emily. This is a sophisticated machine learning platform. It combines structural modeling, it combines generative AI, and what they do is they sift through the whole universe of millions of possible TCR sequences. These are derived from patient samples and from their own microfluidic sequencing platform. And they identify TCRs, potential TCRs, that should bind to a target, an HLA antigen target, based on the structure of that target and then the structure of what these TCRs should be. Emily then uses a large language model. So this is similar to ChatGPT, right? Biology is just language. It's just letters. Bear with me, work with me. So it's using this kind of ChatGPT thing to reiterate and iterate different sequences of proteins based on this original TCR to get ones with higher affinity. And you enrich those, then the important thing is, right, you know, AI is cool, but what you've got to do is prove these things work. They make them in the lab, they test them, and then they put that information back in and continue to iterate until you end up with a very, very high affinity TCR receptor that's suitable for putting into your bispecific therapeutic that should have low cross-reactivity, well, no cross-reactivity, and high affinity. And the first target they've been going after is an antigen called PRAME. Now, this is highly expressed in a number of different tumor types, most melanomas, ovarian cancers, uterine cancers, a significant proportion of hereditary colon cancers, breast cancers. This is a very high patient number population. More than 100,000 patients every year in the US alone are diagnosed with a cancer that expresses PRAME. And that adds up to a $9.5 billion market for use. So this is, and it's also been clinically validated. So this is a target that we know that works in these diseases. And so what they've been doing is they've been iterating through numerous cycles, using Emily to discover a parent TCR that binds to PRAME, and then going through these cycles of iteration, designing, making, testing, putting it back in. And over the course of just 11 months, they've managed to make a TCR that has 11 picomolar affinity. This is absolutely what you need to make a bispecific T-cell engager. There's no known cross-reactivity, and this puts it in the category of being potentially a best-in-class against this antigen. So I thought this was pretty impressive, and so we wanted to tell the world about it. And this is what we did. So we did a media campaign in August 2023, and the aim of this was to take Etcembly out of stealth mode, build buzz around their technology, and support their Series A fundraising round, which is actually shortly to close. And we did it by having two different press releases, one really aimed at the biopharma and tech market, because the AI component is very interesting to the tech world. And we also did put a press release out to the mainstream press, the science journalists, trying to get them interested. We did a lot of personal outreach. We also put it on a newswire. We also provided more background information in the form of a Q&A about the technology, about TCRs, about generative AI in biology, about the company, and we ran a LinkedIn campaign on Etcembly's LinkedIn page. And this is the story we went with. It's a pretty good headline. I'm quite proud of it. And this is the coverage that we got. So we got some really good top-tier coverage, things like article and an interview with Michelle in Inside Precision Medicine, coverage in Forbes, Inc., various other really nice outlets. And also we do have a story coming in a broadsheet newspaper soon, so watch this space. On LinkedIn, we got over 13,000 views in the first week on the story, and we increased their reactions on the company profile by 1,000%, which was really nice. So I think we can firmly say we brought Etcembly out of stealth mode and to international attention. And to summarize, Etcembly is the first company to predict and engineer high-potency TCRs using this proprietary sequence and structural-led generative AI approach. They've, with this same initial asset, they've actually gone on to show that in the lab it's very effective at killing tumour cells against this target. And they're optimizing it and hoping to take it forward for an IND submission. They are available for partnering, guys, so, get in touch with them. And they do have a pipeline of further, what they call ETCs, these bispecific immune engagers in oncology and immune disease, autoimmune disease. Don't ask me about science. If you want to get in touch with the company, that's how you get in touch with them. I'll be around all day if you want to talk about comms. So I will finish there. Thank you very, very much. Vote for us, obviously.
Thank you. Very impressive stuff. So next up, I'm delighted to welcome Amanda Lehmann from Astex Pharmaceuticals for their drug discovery collaboration with MSD.
Hello. So different style of presentation for me, very short, one slide. And no science. My name's Amanda Lehmann. I'm from the corporate development team at Astex Pharmaceuticals. And Astex is indeed nominated for this exciting expansion of this existing great collaboration that we already had with MSD. The aim of this expanded collaboration is to identify small molecule candidates with activity towards P53, which is a well-known tumour suppressor protein, which is why I'm not going to talk to you for slides and slides about P53, because I think the majority of people in the room are familiar with P53. So also the majority of you will have heard of MSD, or Merck as they're known across the pond. So who is Astex? I think maybe more people will be unfamiliar with Astex. So Astex is around for 25 years and is part of the Otsuka Group since 10 years. And they're based in Cambridge. We're on the Cambridge Science Park. Astex has its own structure-based drug design proprietary platform. We're pioneers in this, and we are a leader in this, really. The platform is called Pyramid. Whilst Astex has its own proprietary pipeline of drugs, also collaboration is really part of Astex's DNA. It's really inherent to the way we work, and we feel this is really important for also the success of Astex. We collaborate with people from leading academic institutions. We have the longest running strategic collaboration with a CRH institute, CRUK Institute with Newcastle, and we also partner with Big Pharma, and this is an example, and everyone in between. So as everyone knows in this room, collaborations are really built on strong complementary science and great teamwork, and this is also part of Astex's culture. So Astex has also been successful using this cultural and this science approach with 3 drugs on the market. The 3 drugs on the market is also pretty good for a company that's even at this stage only 130 people. So this new expanded collaboration focuses on multiple forms of P53. Astex will be using this proprietary pyramid platform and will be providing MSD with lead compounds for further optimization and preclinical development. And MSD will, as the big pharma, have responsibility for all future research and development of lead candidates, as well as the commercialization of the products globally. So as you can see here, this is the press release that we put out and some of the coverage that we got from some of the big industry news that I'm sure you're familiar with, Fierce Biotech, Pharma Technology, and Scrip. So why should you vote for this news? It's a drug discovery collaboration. It's an expansion of an existing successful drug discovery collaboration. That also shows that the kind of collaborative culture at Astex is successful, right? If you can grow an existing collaboration, that's a really great endorsement of your internal company culture and science. Collaborations and innovation are really key to success in drug discovery. I think everyone in this room knows this. We've had a lot of discussions about that. So a vote for Astex and this collaboration is also a vote for the pioneering innovation which has placed Astex as a global leader in fragment-based drug design, which allows it to take on challenges such as P53, a challenging target, and also for a company for whom collaboration has always been and continues to be key to its way of working, which is a win for the sector and also ultimately for patients. So please vote for Astex. My pitch is very short because it gives it enough time to get your phone out and vote for Astex on your app. So thank you.
Thank you, Amanda. So Steve Gardner is up next representing Precision Life. He'll be discussing their strategic partnership with LifeArc.
Thank you very much. I'm going for an even more minimalist style because I don't have any slides. In part, that's because of the range of biology that we're going to be talking about. But one of the things I wanted to say, when we talk about the kind of collaborations that we're building, it's quite common for us to use the words patient-centric in the process. When we literally are a precision medicine business that uses analytics on patient data as the heart of what we do. So when we talk about patient centricity, we mean we literally couldn't do anything without patients sharing their data. And we work with some absolutely phenomenal patient groups in wide range of disorders, most recently in ME/CFS and endometriosis. But the patient community that's probably closest to our heart and most impressive is the motor neurone disease and ALS community. These are patients who have been given an absolutely brutal diagnosis, and yet they've found the bravery and the commitment to go out and start sharing their data, which is not going to help them with the best will in the world, but will help other people, help the research community to find solutions for other parts of that patient community. And this is an incredibly impressive group of people. They've come together and organized the collection of data, the funding of large research collaborations. You've seen the Ice Bucket Challenge. You've seen Rob Burrow and Kevin Sinfield running multiple marathons. You've seen the Doddy Weir, My Name's Doddy Foundation, really organizing that community to collect this data. And we are bringing our skills to this. Motor neuron disease is actually quite a personal disease for Precision Life. Our co-founder's brother died of the disease just as we were founding the company. What we've been able to do over the last 6 years, collaborating with King's College and Citran and Motor Neurone Disease Association, is bring a real precision medicine approach to the disease. We found subgroups of patients with different mechanistic etiologies and identified novel drug targets across all of those. They're across different stages of disease, across different cell types. You've heard about some of the target biology, the mitochondrial dysfunction, for example. And there are other classes of targets in what is a very heterogeneous disease. So the collaboration with LifeArc was really part of a national translational challenge to look more widely at all of these different aspects of the disease and try to find solutions for them. This is both in terms of novel target discovery and novel molecular entities. It's also in terms of repurposing. So when we find patient stratification biomarkers for patients and we can connect that target biology to their form of the disease, that also allows us to select patients for whom we can trial existing medicines. And this is one of the most rapid ways of bringing new therapeutic options to patients. This collaboration will actually also lead to better diagnostic tools for the community. And in being able to diagnose patients who should have the disease but don't, we can also potentially find actively protective biology. So it's a very broad-ranging collaboration, and this is a really important part of it. Why should you vote for us? I think one of the things I would love to be able to do is to send a signal to that fabulous, impressive community of people that we as an industry have their back. We're absolutely determined to support them, and we're determined that ALS is going to become a chronic disease, not a death sentence, by 2030. So thank you very much.
Thank you, Steve. Finally, we have John McCafferty from Maxion. Unfortunately, they were unable to join us today, but they have submitted a short video highlighting their $16 million Series A funding achievement to drive antibody development for previously untreatable ion channel and GPCR-driven diseases.
Hi there, my name is John McCafferty. I'm CEO and founder of Maxion Therapeutics. We're delighted to have been shortlisted as finalists for the Genesis BioNewsRound award, and I'm sorry I can't be there with you, so welcome to our new boardroom and our new facilities here in South Cambridgeshire. So Maxion is a company that's been addressing a particularly difficult target class for the delivery of antibody therapeutics. So I was one of the founders of a company called Cambridge Antibody Technology back in 1990. And there's been a real sort of revolution in our ability to make antibody drugs since then. So back then in 1990, there was one single approved antibody drug, and in fact it was a mouse antibody drug with all of the associated problems. At CAT, we developed a technology called phage display that allowed us to make human antibodies to anything we like. That and other technologies, as I say, have driven this revolution such that today there are 180 approved antibody drugs, and in addition there's over 1,000 antibody drugs in clinical trial. So despite all these successes and advances, there's one particular target class that just remained intractable to antibody therapeutic development, and that's the group called ion channels. So ion channels are signaling molecules on the body. There are hundreds of them involved in normal health, but they are also associated with a number of pathologies, pain, ischemia, autoimmunity, and so on. It would seem like a good idea to make antibodies to these drugs, but despite the efforts of biotechnologists over the years, there are still no approved antibody drugs that address ion channels. And that's really what Maxion is about, to try and solve that problem. So we're actually trying to learn a lesson from nature. So unlike the efforts of biotechnologists, nature provides these small cysteine-rich peptides that have been shown to have ion channel modulating activity. They do not themselves make great drugs. They've got very short half-life, difficult to engineer and produce. So really the idea here at Maxion was to combine the best of both worlds, to take these small cysteine-rich peptides and embed them into the surface of an antibody, and thereby enjoy all the benefits of antibodies: long half-life, engineerability, standard manufacturing, and so on, but now souped up, if you like, with these nature's ion channel modulating scaffold. So we call those molecules not bodies, and we've been able to generate not bodies to several therapeutically relevant ion channels. So we've generated news on a number of fronts this year, and in particular there was a press release went out in February of this year announcing the completion of a seed fundraising where we raised 13 million in a round led by LifeArc, but also including BGF and Monograph. And very shortly after that we also announced a success in a 2 million Innovate UK award competition. So we're really using the money from that seed round and from the Innovate UK to move forward with our dream of generating antibody-like molecules to address this important target class. And we look forward to using the technology and using the funding to drive us towards the goal of making new medicines addressing this untapped opportunity for improving human health. So I hope you will agree that this is an exciting story and will vote for Maxion Therapeutics. Thank you. Goodbye.
Wonderful. So thank you to all of our speakers for your excellent pitches and good luck. Now it's over to you all in the audience. If you haven't already voted, please do so by the app. With that, please do go and enjoy your lunchtime. We will be back in here at 1:20 with Alexandra Gray for What Would I Like from the Life Sciences Industry. Thank you.